Hello everyone, and welcome back to the series on the immune system. Today we are going to go through section 3 of the chapter, which is on adaptive immunity, defense against infection of bodily fluids and bodily cells. So, in the first section, going back to the second video, we have defenses based on the B and T lymphocytes, and we have it divided into two separate sections. We have the humoral immune response, which is based on B cells, activation of B cells. Immune response, which is determined by the activation of T cells. It loads. Cytotoxic T cells, defending against infected cells. Now we got a lot to get through in this video, so we're going to leave it at that. We'll talk more about it, but on to helper T cells. Now, helper T cells are a special kind of T cell that secrete cytokines to promote the two responses we just talked about, the humoral and the cell-mediated response. We'll see how this works here in figure 43.16 when we get to it. But first, we'll talk about the requirements that must be met. So, first, a foreign molecule must be present that matches the antigen receptor on the helper T cell. Secondly, the antigen must be present in this antigen presentation that we had talked about previously in the series. So the major histocompatibility complex has to grab an antigen fragment, and it must be presented, and it must fit to the helper T cell. The cell presenting things, so the MHC molecule, can be dendritic, which we had talked about, present in skin cells, macrophages such as white blood cells, or it could also be a B cell. The book also describes that we have two different classes of MHC molecules, as you can see here in the figure we're going to look at. There's a class 2. Most body cells only have class 1, but antigen presenting cells have class 1 and class 2, and the difference is that class 2 molecules provide a molecular signature by which the antigen presenting cell is recognized. Now, before we move on to the next page, let's take a look at this figure. So this is the role of helper T cells, as you can see in the description. In the first picture, we have the endocytosis of the pathogen by a host cell, and it degrades it, presents it. The helper T cell then binds to it and secretes cytokines, which then initiate the humoral and cell-mediated responses from the immune system. So the next page actually has more information about this. So when we go forward, we can see that this section pretty much describes that there's an accessory protein called CD4, which binds the class 2 MHC in the T cell, and this binding lets the cytokines be produced, and dendritic cells and macrophages activate the T cell, which proliferates and clones, but B cells present antigens to already activated helper T cells, which allows the B cells to activate as well. As it says here, we'll move on to cytotoxic T cells. Now, the cytotoxic T cell section that we have here is going to be similar to the helper T cell with a couple differences. So, cytotoxic T cells destroy infected cells, as well as certain cancer cells, to stop the production of pathogens. So, the book shows you here's an infected cell with pathogens reproducing and coming out of the cell and using it as a good environment to reproduce. So cytotoxic T cells come in and kill the infected cells before the pathogens can mature. So like helper T cells, cytotoxic T cells have to have an antigen receptor to match the presented antigen, and they also have an accessory protein like the helper T cells do, but For cytotoxic cells, it's CD8. Sorry about that. So the job of the cytotoxic T cell is to stop the production of the pathogen. And down here in figure 43.17, we see the killing action. So just like in the helper T cell, it connects, and then we get a release, sort of like cytokines with the helper T cells, but instead we get the release of perforin, which perforates the cell membrane, and then these granzymes can enter the cell and perform apoptosis of the cell in order to stop the production of the pathogens. So that's a pretty good overview of the T cells. We're now going to move on to B cells and antibodies. So again, like the cytotoxic T cells, this B cell section has a lot of similarities to the helper T cells to infer to what we learned initially. So secretion of antibodies by B cells is the hallmark of the humoral immune response and begins with the activation of B cells. So as we'll see in the figure when I go through it, the activation involves T cells and proteins. So here the book just specifies that B cells can only present antigens to which it specifically binds, as opposed to macrophages and dendritic cells that can provide fragments from a wide variety. So what we're going to get here, we're going to go to the figure now and discuss everything on this side afterwards. But this is just a representation of what we had learned initially with the helper T cell. You have the accessory protein, that class 2 MHC molecule. The receptor has to fit here. This has to be presented. Pathogen comes in and is engulfed by the antigen presenting cell. And then the helper T cell emits cytokines, which it does not show here. But this is what we learned initially, and this is the same thing, except with a B cell, when the cytokines are produced, you get the activation of this B cell, and the B cell proliferates, sorry, I think it said pour. Like how we had discussed earlier in the series, it clones itself into memory cells, which are used for long-term usage, and plasma cells, which differentiate to secrete these antibodies. So the plasma cells secrete approximately 2,000 antibodies every second during the 4 to 5 days that they are used. I skipped over a single activated B cell can clone to give thousands of plasma cells. So a single activated B cell produces 1,000 plasma cells, which produce 2,000 antibodies every second. So each plasma cell produces nearly 1 trillion antibodies in total. It also specifies that most antigens recognized by T cells contain multiple epitopes. So exposure to a single antigen activates a variety of B cells, give rise to different plasma cells producing different antibodies. So you get a large amount of antibodies produced from a single pathogen. So on to antibody function. The book specifies that antibodies don't actually kill pathogens, but in a variety of ways they hinder the pathogen itself. They interfere with work or mark them for inactivation. So the first process we're going to talk about is neutralization, which is on the next page. You'll get a figure. The bound antibodies prevent infection of a host cell and neutralizing the virus. Similarly, they bind to toxins released in body fluids to do the same thing. So figure 43.19a shows neutralization. The antibodies bind to the epitopes, which they can, to block the virus's ability to bind to a host cell. So, sort of like as we had talked about for my fellow classmates, we had talked about in class inhibitors. This is inhibiting the virus's ability to bind to a host cell. And figure 43.19 section B talks about opsonization, which is described below here, if you guys are still following along in the book. The antibodies bind to the surface of these to tag the bacterium or the pathogen for phagocytosis by a macrophage. And so, because the antibodies have two binding sites, they can also link together bacteriums, and they can also they initialize the rest of the humoral immune response. So because they're initiating phagocytosis, it can also enable the stimulation of helper T cells and B cells. So it says this positive feedback between the innate and adaptive immunity contributes to a coordinated, effective response to an infection. And up here in the third section, we see activation of a complement system and pore formation. So it's described down here. Antibodies can sometimes activate this complement system, which forms pores in the cell membrane, and water and ions flow into the cell and make the cell so turgid that it breaks apart and is no longer functional. So the antibodies in this way can initiate apoptosis. So down here, the book talks about antibodies causing death of infected cells, B cells, and their expressed types. And then here we get a discussion about the responses enabling secondary response. So the memory cells, as we talked about earlier, upon reactivation here, initiate a secondary humoral response, which we already know about secondary responses. So this large figure on this page, we're going to get an overarching view of adaptive immunity, which we've been talking about for the past two sections. And we're going to take a quick look at this before we come down here and talk about active and passive immunity. So, as we've said in this section, we've got the two different sections. You got humoral response, cell-mediated response. We get a first exposure. We come over here, stimulation of a B cell, or for both sections you get something like a macrophage engulfing the antigen, and then presentation to a helper T cell, or presentation to a cytotoxic T cell. These helper T cells can activate B cells, and this line represents proliferation into plasma cells and memory B cells. Plasma cells secrete antibodies. You can activate memory helper T cells upon a second exposure. You make these. You make memory B cells, and you make memory cells in general for the second exposure, so that you can have a quicker recovery time. And then finally here you get cytotoxic T cells. Down here, the active cytotoxic T cells. They defend against intracellular pathogens and certain cancers by lysing, killing the cells. And over here you get antibodies, which defend in the ways that we had discussed earlier on this page. Back here you get neutralization, opsonization, and activation of complement system and pore formation. So I'm gonna leave this figure open for a while, so you guys can pause on the video, study if you need to, because this is really a good overarching figure. And I'll come down here a little bit, so if you guys want to pause on the video, go back and read this, study it, take notes on it, I would suggest that's a really good place to for kind of a summary of what we've been talking about. Now, on to active and passive immunity. As it says, we've been talking about active immunity, which is long-lasting immunity conferred by the action of B cells and T cells. So now we're going to move on to a new subject, passive immunity. Now this is a very specific type. It results when one of the 5 types of B cell genes produce antibodies in the blood of a pregnant female. They cross the placenta to her fetus. So, passive immunity, short-term immunity conferred by the transfer of antibodies. And it says they're called this because there's a recipient, in this case the fetus, of antibodies which are produced inside of another individual. Another example is IgA antibodies present in breast milk are also passed on to an infant. And because the passive immunity does not involve B cells and T cells, there's no long-term effects. It only lasts as long as the transferred antibodies last. So as long as a fetus is in utero, for the example of IgG, or for as long as breastfeeding is done for IgA. So on to the next page. So in this section immediately talks about active and passive immunities being induced artificially. So immunization is an example of active immunity. So when I think of this, I think of an inoculation. For I think probably the most common inoculation is influenza. When you go to get a shot, they actually they introduce a inactive or weakened form of the pathogen so that the responses are sort of get a head start. But it's like it says, it's temporary protection. So for active, you're sort of jump starting the system in a way. And this section just sort of gives a history for it. You can pause and read it if you want, but it's not really as important. Already at 18 minutes, so trying to hurry. So this section really just talks about the successfulness of vaccination programs against infectious diseases such as polio, measles, whooping cough. Really, again, it's like a history thing. If you want to read it, I'll leave it there so you can pause it. Artificial passive immunization. Antibodies from an immune animal are injected into a non-immune animal. So the example it gives is snake venom. When someone's bitten, serum from sheep or horses is injected into the person, and if it's fast enough, the antibodies can neutralize the toxin before they do massive damage to the person. And this talks about antibodies being produced. Antibodies can be prepared from a single clone of B cells grown in culture. So that's what a monoclonal antibodies are. They're all from a single origin, a single B cell. And they're all produced in culture for a specific epitope on an antigen. Sorry. These monoclonal antibodies have been proven useful. They're used now a lot for home pregnancy test kits to detect human chorionic gonadotropin, maybe? I don't know. So that's my guess. Anyway, sorry. So the monoclonal antibodies can be useful. This section talks about how things such as for passive immunity, parts of another organism can be rejected by the recipient organism, like skin grafts, blah blah blah. Really, what I found interesting about this is this this section that says it remains a largely unanswered question why a pregnant woman does not reject her fetus as non-self. It's I don't know. It sounds like a cool concept to me. That is a good question that hopefully young adults like myself and maybe the people watching this is the kind of things I'd like us to figure out. That'd be really cool to know. Moving on, as we also talked about class, we talked about blood groups, but we talked about this in the sense of dominant and recessive alleles. So you have the 4 types of blood. They have to have the right alleles, consistent, so that blood is not rejected by a recipient. This continues talking about the 4 types. This section talks about how if you have different blood types added together, how the antibodies react. So here, if a person with blood type A receives a transplant of B, the anti-B antibodies in the type A blood start to lyse the red blood cells, which cause a lot of painful and very bad side effects: chills, fever, shock, kidney malfunction, and then same thing vice versa. In this, so O-type blood doesn't have any of the epitomes that A and B do, so A epitomes are rejected by someone with type B blood because the antibodies connect to the A to destroy them, and vice versa in B, but O doesn't have any of those, and type AB has both of them. So O cannot accept A or B, but A and B can accept O, A, and B, which is why you get AB is a universal recipient, O is a universal donor, because everyone can accept O, and AB can accept everyone. This section talks about tissue organ transplants, the use of the MHC molecules. Physicians try to get MHC molecules matched as closely as possible so that there's no rejection. Also, it notes that recipients take medicines to suppress immune responses, but in a result, their immune system is weakened. Specifies that bone marrow transplants are different because you can have an immune reaction, but instead the B cells are eliminated out of the patient's own bone marrow, so to destroy the source of abnormal cells. It obliterates the recipient's immune system, leaving little chance of reaction. However, the lymphocytes in the donated bone marrow may react against the recipient. So again, that's why physicians try to match that MHC molecules very well. So, like I was saying, very nostalgic, very cool concepts being introduced. Next video will be the last one. Part 4 will be on disruptions that can elicit a disease or make it worse. So I'll see y'all then, and take care.